Thanks for the introduction. I'll switch to English since we have some in the room who are English speaking. And yeah, I would like to welcome you to my presentation about postural control mechanisms. And I would like to start my presentation with a question why it is actually interesting from a sports science perspective to investigate and work with postural control. And when we talk about postural control, and especially stance, we talk about a basic human ability that we all learn in quite an early stage of our lives, as long as we are healthy. And although it is a very basic human ability, it is a very complex one. And that is due to the joints, muscles, bones that are involved into these motor tasks. And we still do not really understand what is happening in certain situations. And one fact is also that it is largely automatized, meaning that, for example, I can stand here and talk to you without consciously thinking about standing at that moment. And how these automatisms actually work is still topic to debate, especially in motor control research. So we don't know exactly how these automatisms work and what are the mechanisms behind. And coming back to my question, why is it interesting for sports science? Well, if we better understand such a basic human ability, how that actually works and how the motor control system reacts to certain situations, we might be able to give implications for training and rehabilitation afterwards. So when we talk about analyzing postural control, we always have that degree of freedom problem that some of you might have heard already. As I told you, we have a lot of muscles and bones involved in movements, and so we have a lot of possibilities to actually perform movements. And to analyze these movements and the postural control, we have to reduce the dimensionality. There's one example, for example, having just a focus on the ankle joint, being one joint that is very active during stance, and to try to analyze just that one single joint as one dimension in the whole context. That would be the single inverted pendulum. But as you know, we have more than just the ankle joint, so we could add another dimension and also have a look on the hip strategy. Then we have already two dimensions, having a double inverted pendulum, but still it's not covering all of the information that actually is active during stance or during postural control. And we have another possibility to measure postural control, for example, using center of pressure as the resultant ground reaction force that we project on the ground when we are standing. And you see here, there's quite a lot going on, even during stance. So when we are standing, we are moving all the time. Our agonists and antagonists, the muscle synergies are working. And again, center of pressure is another variable, but again, just two-dimensional. So we bring everything, every information into one global measure of that whole complex situation. When we talk about center of pressure, we at least have we know that it actually correlates quite well with the inverted pendulum models I just showed you. And we also can use the center of pressure to analyze and to see effects on postural control. So for example, in dual tasking, we know that the center of pressure sway decreases. So during dual tasking, we sway less than when we focus on stance. In adolescence, for example, we also know that adolescents, although they appear to have a similar postural control system than adults, they also sway more than adults do. And another example for using center of pressure as an analysis method is after neurological impairment. So after concussion, the regularity of the center of pressure decreases, so it becomes more regular after concussion. The problem now with center of pressure is, as I told you, it's a two-dimensional global measure. It's very hard to explain the underlying mechanisms that are active and that actually behave and work together. So the mechanisms that could be an explanation, but we trouble with actually analyzing or explaining with center of pressure is the optimal feedback control theory. Now, very complex word, or maybe it's not easy to follow what that means. Basically, it means to use feedback that we get when we perform movements in an optimal way. And one part of that optimal feedback loop, so to say, is the minimal intervention principle. And the minimal intervention principle basically says that when we move, we get feedback from our sensory system. And our optimal feedback control law now has a lot of dimensions. As I told you, we have a lot of joints, a lot of possibilities to behave. There are many movement dimensions to control and that could work. And the MIP now thinks that some movement dimensions are more task relevant. For example, during stance, that ankle strategy that I also showed you in the single inverted pendulum. And some that are not as task relevant. They play a role, but they are not as task relevant to the overall movement. So for example, here, the breathing movement. That plays a role, but it's not as relevant as for example this ankle strategy. And we found this MIP, for example, in muscle activity patterns. We also found it in joint torque patterns. So it seems to hold true. There seems to be quite a reason to think that the MIP is active in our postural control system, but we still don't know how it is when we talk about adjustments to postural control. So does it also hold true when we talk about adjustments or altered sensory motor integrations? Is the MIP a possible explanation there as well? And also how to analyze posture with focusing on such a high dimensional context. And that's exactly where we have to first increase the information we gather, and then again to decrease the information we gather to actually be able to analyze it. And when we talk about increasing information, for example, not only having a look on the center of pressure, but on more information, we can take our participant and use motion tracking. So we take 39 reflective markers, place them on anatomical landmarks on the body. Then our participant looks like that with the markers on. And then we take 8 cameras which see these markers and which record their movement basically, so of every single marker. And then our participant will light up, hopefully, and we get the information about the coordinates of that markers. So now we have a lot of information we can work with, but how to analyze. And that's where the PCA comes into play. And the PCA basically brings in all of that information in one big data matrix after it's been centered, weighed, and normalized. And then it calculates which movement directions or which data directions have the largest share to the overall variance. And so we then get, for example, the eigenvector 1, which presents the largest share. We also call that PC1 or PM1. And then the second largest share would be PC2, and so on and so on. So the higher the number, the less share it has actually, and always continuing to increase. And then we get different movement dimensions, so to say. So we have, for example, in tandem stance, a mediolateral ankle sway, which is responsible for 60% of the variance during tandem stance. And the second largest share of 25% would be an ankle strategy in the anterior-posterior direction. And so the movement basically gets cut down to the most important movement dimensions and going to all movement dimensions that play a role. We get it as an output the eigenvalues, the eigenvectors, as I told you, and the scores. And the scores we also call principal positions, as they represent the movement where the markers basically go, and that in the context of the kinematic marker data. And we can differentiate these principal positions, and by differentiation we get the principal velocities, and we can do this again, and then we get the principal accelerations. And in the accelerations, we think that it is a very interesting variable because every time the system decides to change the acceleration, the direction of the acceleration, we think that the postural control system acts to bring a neuromuscular control in there. And if we count these number of direction changes, we get the so-called number of zero crossings. And they basically tell us, all right, do we have a very tight postural control? So is the control very high when we have a high number of these zero crossings? Or is the control not as tight, so a little loose, and the control is not so tight because of not as many number of zero crossings, so not as many direction changes. We also can calculate the relative variance out of the PP, and that basically tells us how much share does each movement component has to the overall movement. So it tells us something about the coordinated structure of the movement of, for example, tandem stance as we had in the example. And we also can calculate the absolute variance, and that tells us how much sway or how much amplitude each of the principal movements has and how it actually behaves. And we also, that's the fourth variable I was aiming at, we can also calculate the entropy. And entropy basically is the level of the regularity, and regularity is also about predictability. So how easy is it to predict where the next point or the next time series point is going to be. And the higher the value, the less regular the movement is. So these were the four variables, and my research question was with these four variables to tackle the question, all right, which mechanisms does the postural control system actually follow when we talk about an altered sensory motor integration? And I had three different situations. First, I had dual tasking, I had adolescence, and I aimed at head shaking and how the postural control system behaves after a head shaking. And I want to start with the dual tasking. As I told you, we know that center of pressure, for example, decreases in sway during dual tasking. And the question was now, why is that? Why is that actually happening? Is that due to active automatisms? Is that because the neural system deprioritized or actually prioritized differently? And overarching, is the MIP maybe an explanation to explain what is happening during dual tasking compared to single tasking? And what we did was we analyzed 38 participants, 23 were adults, and 23 were adolescents, and 15 were adults. And we had three conditions. We had a single task where they were standing in bipedal stance with eyes open. We had another single task where they were standing in the very same position but with eyes closed. And we had a dual task where they were standing with eyes open but they were counting backwards in steps of 2. And these three conditions were analyzed during stance for 25 seconds. We analyzed the center of pressure and the PCA, where we had a look on the eigenvalues, the number of zero crossings, and the absolute variance, so how much amplitude has each single movement dimension. For the statistics, we tested, of course, for normal distribution, did a repeated measure ANOVA for the 3 different trials, and we also did a Holmborn-Ferroni correction because you will see we had quite a lot of movement components, and to kind of avoid an alpha cumulated error, we also did a Holmborn-Ferroni correction to have a more conservative statistical model. If not normal distributed, we did a frequent test. And the results show that, for example, the ankle strategy we were talking about already plays a significant role. I mean, it's almost 64% of the overall share to the movement. And another dimension you already know is the hip strategy we were talking about. And that's, for example, PM number 3, also having a relevance in that movement of bipedal stance. And when we now have a look on the amplitude of these single movement dimensions, we can see that in the first ankle strategy movement dimension, we have a significant decrease between the 2 single tasking trials and the dual tasking trial. So in the dual tasking, the sway in this particular movement component is not as high as in the single tasking trials. Compared to the other components, we have a totally different pattern, interestingly. So in all other movement dimensions, we have an increase during the dual tasking. So all other movement dimensions presented an increase in sway when we are talking about dual tasking compared to single tasking. When we talk about the number of zero crossings, so the neuromuscular control, we have a different opinion or a different pattern in the first movement component. We have again significant differences, but this time the eyes closed trial appeared to play a significant role. But again, we had the same pattern in all of the higher movement components. So all movement components ranging from 5, 7, 8, and 9 presented this time a decrease and all with the same pattern. Now you might have noticed that we also had a look on adolescents and adults, and there actually were no differences or not many differences between these two groups. So basically we think that maybe during bipedal stance it's just too easy, so there is not a real difference between adolescents and adults. How can we in general discuss our results? Are they, as I said, active automatisms? We think yes, maybe not at first glance, but if we have a closer look, we see that there is a pattern. So we have the first movement component, the most important one, having 64% of share to the variance, acting different than all of the other movement dimensions. And that might be due to a different prioritization of the neural control system. And is the MIP now active? Well, since we have one movement dimension being maybe more relevant than the others and therefore behaving differently, we would say yes, it is actually active. Now, dual tasking was a success with our hypothesis, so to say. What about the adolescents? Do we have the same kind of results there as well? With the adolescents and adults, we wanted to know, do they actually control difference? So do adolescents and adults control in a different way? And if they do, or maybe on top, is the coordinative structure different? So do adolescents use different movement strategies than adults? And also again, overarching, is the MIP active? Is it an explanation of what is happening there? In the adolescence trial, we had 26 males, 12 were adolescents, and 14 were adults. And to also check for a body height effect, because in literature it's often argued that maybe body height might play a role in different movement strategies or coordination, we divided the adults into 2 subgroups. So we had one group of small adults with being under 180, and another group of tall adults being over 190 of body height. We had this time two conditions, both in tandem stance, because this time we wanted to have a little more of a challenge for the postural control system. And tandem stance, as you see, is not a typical daily standing position, so to say. And we had it with eyes open and eyes closed. And you can read here just pure stance in 16 seconds. And that is because actually the adolescents, for example, really had trouble to stand without any compensatory movement. Meaning that sometimes they were standing and lifting one toe and therefore kind of compensating a fall. And we totally cut out these kind of compensatory movements and just focused on pure postural control with successful postural control. We again did a PCA, eigenvalues, so how much share of each component, number of zero crossings, and this time relative variance, because we wanted to know, do the movement patterns change? Do we have a change in coordinative structure? Normal distribution tested, then repeated measured MANOVA to have every single variable in a context together, and we had three groups, so that's why the MANOVA, and subsequent t-tests, if not normal distributed, Crossfield-Wallace and Mann-Whitney for the postdoc. Again, Hornbaum-Ferroni, because again we had 9 different movement dimensions, and you see something similar than before. So you have again an ankle sway as the main strategy, also during tandem stance, this time in mediolateral direction, and also the hip strategy in mediolateral direction, so that kind of goes in line with what we saw after the bipedal stance. For the adolescents, and compared to the adults, there was no change in the coordinative structure. So the relative variance could not show any significant difference, meaning that the movement strategies, so the share of each movement component, remains rather unaltered. On the other hand, in the number of zero crossings, we found significant results, not in the first component this time, but for example in the second component. And interestingly, it's between the adolescents and the small adults. No significant difference between the adolescents and the tall adults. And this pattern actually was true for all other movement dimensions, and we also could see 3 more movement dimensions showing the very same significant difference between adolescents and the small adults. So how can we now discuss these results? First of all, do adolescents control differently? We would say yes, because we have a significant difference in neuromuscular control, talking about the number of zero crossings. So adolescents control their movement dimensions differently. Is the coordinative structure different? We did not see any significant difference, so we would not say that the movement strategies are different. But is the MIP now active? Well, if we have a closer look on the movement dimensions that were actually different, we see that it were just movement dimensions that represent anterior-posterior direction. So now you have to imagine when you're standing in tandem stance, the anterior-posterior direction is not as vulnerable to lead to a fall than the mediolateral direction. If you make a mistake in the mediolateral direction, it might lead to a fall way more easy than when you are in the anterior-posterior direction. And we discussed that maybe the adolescents try to find their boundary condition in the anterior-posterior direction, because the system knows these movement dimensions are not as task relevant, so there we can explore and find our boundary condition. All right, now we have the adolescents, and perfect transition to headshaking, because I guess a lot of adolescents look like this in their wild times when they find their new music. And the headshaking trial, that was the idea. We know from center of pressure that it behaves differently after concussion. And now we wanted to know, do we have the same event, the same result when we do a headshaking? And also when we see the same pattern, is that due to an altered temporal structure? Is that due to a different coordinative structure? So again, do the movement strategies change? And is the MIP a possible explanation for the results that we might see? This time I want to start with what is regularity when I talk about regularity. Here you see a perfect example of a very irregular time series. It's very hard to predict where the next point of that line is going to be. So you would talk about an entropy level of 2, so a high entropy. And the very opposite would be a sin wave, where it's very easy to predict where the next point is going to be, and that would be a low sample entropy of 0. So these two are important to know, but I will come back to that. And interestingly, if you are healthy, so you are not impaired at all, you will show a more irregular center of pressure movement than when you are concussed. After concussion, your movements become more regular, at least when we talk about center of pressure. So what did we do this time? We had 16 healthy participants. We calculated the center of pressure and again the kinematic marker data. And they basically stood still in bipedal stance for 30 seconds. Then we gave them a command to start the head shaking, and the head shaking looked like this in a nodding movement, or like this in a tilting movement. And we did both trials with eyes open, eyes closed, so we had 4 trials in total that we had to look at. And after 10 seconds, we told them, all right, stop. And they stopped their head shaking as long as they heard us, and they stood still for another 90 seconds. So we calculated on that data from the center of pressure the sample entropy, and as well on the principal positions. And we also had a look again on the relative variance, so does the coordinative structure maybe change after such a head shaking. And this time we did a repeated ANOVA again, if not normally distributed Friedman. No Horman for only this time because we just analyzed 5 movement dimensions. I will just show you the result of the tilting head sideways with eyes closed because they represent the result that we saw the best. And when we have a look on the center of pressure, we see that in the phase 1, so before the head shaking, we have a rather high sample entropy, so irregular. And immediately after the head shaking, so the head shaking occurred, and then immediately after that stopped, we took the second phase, and there you see a decrease in the entropy. So basically, the movement became more regular. And then the third phase was immediately after the second one, and again you see a significant increase in the sample entropy. So exactly what we wanted to see, because that's exactly the same pattern as we know it from concussion. So now answering, is that also the fact for the principal positions? And yes, we see the very same pattern in all of the movement dimensions, and especially in the tilting the head trial with the eyes closed, we saw this decrease in regularity after the head shaking. So how can we discuss these results? Is it a similar effect, head shaking concussion? It appears like that, yes, at least when we talk about center of pressure. And is that due to a temporal structure? We would say again, yes, because the principal positions, so the movement components that are very important in standing, they show the very same pattern than the center of pressure. Is it due to an altered coordinative structure? Well, we also had a look on the relative variance, as I told you, and here we see the cumulated relative variance of the very first 5 movement components that we analyzed, and there were no significant differences. So after the head shaking, the movement strategies remain rather unaltered again. This time, MIP active, hard to tell. Since every single movement component presented the very same behavior and pattern, we would say this time the MIP is not a good explanation because we do not see any difference between task-relevant or task-irrelevant dimension. All right, after having these 3 trials done, what can we conclude? What is the general discussion? I want to start with a research question again. So what are the mechanisms? What is the underlying mechanism after an altered sensory motor integration? First of all, the coordinated structure of our movements seem to be rather unaltered. So we have our movement strategies, the movement components have their share, and that does not change. There is no difference. On the other hand, when we talk about the minimal intervention principle, it seems to be quite a good explanation for what is happening in dual tasking and adolescence, and that is because just the task-irrelevant dimensions showed a significant difference and explained or behaved different than the task-relevant dimensions, and that's exactly what the MIP basically says. After head shaking, we cannot say that the MIP is true or holds true, and we think that maybe there is a change of relevance in the movement dimensions after such a perturbation like head shaking, or maybe the system simply fails to distinguish, all right, which movement component is now relevant and which is not. Or at least we could not see any explanation or any result that would support the MIP in that case. Also, I told you that posture and postural control is largely automatized. And maybe these alterations, so these altered sensory motor integration, leads to, or affects the automatisms that we have in our system and that work very good. I mean, I'm still standing, I did not fall so. And if we now talk about what the results could mean, if we talk about stance and we have a cognitive task, so we are not focusing on our standing, but just on the task. We have a cognitive resource competition because we need to stand and also answer a question or, for example, count backwards. When we have an altered sensory motor integration on the other side, we see that the sway in the cognitive task, that is what we know from the center of pressure, is less than during the focus on standing. And why is that? And there we think there might be a change or a difference in the higher and the low order movement components. So when we talk about the cognitive task, we saw that, for example, the amplitude in that very lower order component, being the ankle strategy, decreases. Maybe that is already an automatism that just focuses on this one very task relevant dimension and tries to control it in a way that it is not as much sway. On the other hand, when we focus on standing, we try to stand as still as possible, and we maybe are not as good as our system. So in that very first lower order component, the ankle strategy, the amplitude increases, and that might lead to this general increase of amplitude or of sway in the center of pressure. In the higher order components, we saw a different behavior. So we had an increase of the absolute variance, meaning that maybe the system knows, all right, I have my first movement component perfectly controlled, the others can go up a little bit in amplitude. On the other hand, the system also knows that we try to kind of consciously control the first movement component, and then it just reduces the amplitude in all the other trials as we saw, in all the other movement components as we saw, to keep us standing in sake of a little more sway that will not lead us to fall, but it is there. And these two results, or this explanation, also goes in line with the results that we had in the higher order components with the number of zero crossings, because where the amplitude went up, the control went down, and where the amplitude went down, the control went up. So actually it's pretty much in line with what we saw. And that is again in line with the general idea of the MLP, having some movement dimensions, task relevant, differently controlled than the not so relevant dimension. And this hierarchically levels of the movement dimensions should be considered in future research because they seem to explain some of the mechanisms that are active during, for example, stance. And it would now be very good, and I would appreciate in the future if we would see intervention studies to see, is the MLP, for example, trainable? Is there a possibility to see how it behaves differently after different training strategies? And also to give some practical implications out of my results. First of all, I would highly suggest to establish automatisms by repeating movements and giving the system the ability to learn movement, but then also to experience various movements. And I'm really much convinced by the differential learning theory there that it's very important to know or to bring the system into situations where it has to learn new systems or new ideas how to behave in a certain situation and to just be able to adapt. And also to support the automatisms, it might be a good idea to train muscles in a different manner, because we do not only want to have strong muscles, we also want to have very good coordinated and neurological, perfectly working muscle synergies. So it would be a good idea to do that in a diverse manner in training of muscles. The limitations, well, the sample sizes were not huge, as you saw, especially after cutting them into different subgroups. Nonetheless, the significant results were quite good, and we also had nice effect sizes. So I'm pretty confident to state that the results that we saw are valid. And another limitation goes to the principal component analysis. It is just a linear approximation of the movement. So these components that you saw are not real movements, but they are linear approximation. They still give us an idea, but we cannot say, all right, that's now the ankle movement, so to say. It's just the representation. Coming to an end, I want to thank, first of all, the Neurophysiology Research Group I was working at the last couple of years. It was a pleasure. And also to all of my co-authors, of course. Especially I want to name Peter as my supervisor. Thank you for your guidance and for your support. Thomas, for really helping me to get into this PhD thing and helping me with some issues I had in the very beginning. And also to the two guys which are new in the group, Maurice and Stephen. It was fun work with you in the last couple of weeks. So Stephen, thank you well. And also I want to thank the whole Institute of Sports Science. I really felt appreciated all the time here. I did now almost all of my studies here, not almost, but hopefully almost when it's positive after that. And I enjoyed working here, and thank you all for the positive atmosphere at the Institute. I want to thank my family, especially my parents, for always supporting and saying you do a good job. It really helped. And last but not least, of course, Steffi, thank you for all your support, endurance with my foibles sometimes, and just want to say thank you. And now I want to thank you all for your attention, and I'm happy to answer some of your questions. Thank you.